Welcome to our update from day 3 of the Keystone Symposia Conference on Neuroinflammation in Diseases of the Central Nervous System. What I'm going to talk to you about today isn't so much the data that we heard in the talks, but more a concept that's really important in neurology. And so one of the sessions that we had today was specifically about the blood-brain barrier. So while everyone who works in the field or has an interest in the field probably will have heard about the blood-brain barrier before, it's often a concept that you know about without necessarily knowing all of the details of it. So the first thing that I want to talk about, and this was really nicely summarized today, is what's different in the blood-brain barrier compared to blood vessels around the body. So the main difference that we know is that the blood vessels in the brain, the vasculature in the brain, is a lot less leaky than blood cells around the rest of the body, than blood vessels around the rest of the body. The easiest way to think about that is if we were to take two balloons, one representing blood vessels around the body and one representing blood vessels in the brain, and fill them with a coloured dye, put the balloon into water, what you would see is that from the balloon that represents the rest of the body, some of that dye is going to have escaped out, passed through the balloon and into the water over time. Whereas for the balloon representing the brain, because of this blood-brain barrier, all of that dye will have stayed inside the balloon, so the water will still be clear. So there's this main difference here that it is less leaky, but why is it less leaky? There's a few different things that contribute to it. The first thing is that the cells that make up these blood vessels in the brain are a lot tighter. They have these things called tight junctions, which essentially bring them closer together. So there's less gaps between the cells. So there's this physical nature that makes them less leaky. As well as this, there's less movement of cells across from within the blood vessels out. There's also less molecules that immune cells or cells of the immune system can stick to, which is an important feature around the rest of the body. These adhesion molecules are what the cells of the immune system will stick to and then migrate into tissues, so there's less of those molecules for them to stick to. And the other main difference that these blood vessels have in the brain is that they have specific transporters or ways for certain molecules to get out, because obviously the brain, for example, needs sources of energy such as glucose. So because of the fact that these blood vessels aren't leaky, there's specific transporters to help those molecules get out of the blood vessels. So that clarifies what the blood-brain barrier is. But what I want to talk about, and this is the only data that I'm going to talk about today, is we're interested in the blood-brain barrier because of the fact that we know that the breakdown of the blood-brain barrier contributes to MS, epilepsy, brain trauma, stroke, but we don't really know what processes are happening to cause that breakdown, or whether or not the breakdown is the same in all of those different disorders. So obviously the different disorders that we just mentioned have very different outcomes and are very different, but what this research group from the University of California have found recently is that the mechanism by which the blood-brain barrier breaks down is very similar across all of those different disorders. And they even think that it may just be the mechanism by which blood-brain barrier breakdown occurs. And that's really interesting because the fact that immune cells can get into the brain because of this blood-brain barrier breakdown is one of the key pathological features of say, a disease like multiple sclerosis. So being able to understand what causes the breakdown of the blood-brain barrier is really important, because if we can reverse that breakdown and make it less permeable again, that can be a way of potentially helping to treat and improve the outcome. for some of these conditions.